So all of you guys probably sort of know why we're here today. Vighnesh has done his master's recently, ended up having to do it via, virtually due to the COVID situation. He was going to do his master's on sharks, but through a random Facebook connection, ended up in contact with Chad, my boss at Conservation Diver here, who they sort of got into discussion of mineral accretion technology, and Vighnesh has ended up doing his master's on that. And in doing so, he's written what we think is a pretty incredible literature review. Chad was so bold as to say he thinks it's one of the most in-depth ones that he's ever seen, which I promise you is quite a large statement. So he's accumulated quite a wealth of knowledge along the way, of course, and that's why he's presenting today. He's also volunteered in a variety of different positions within the marine realm through different organisations. And he's actually just about to start yet another degree, doing his Masters of Science in Marine Biological Resources through the Erasmus Mundus Scholarship of the European Union. So well done for you for managing to be willing to put yourself through yet another master's program. I'll pass it on to you now, and you can, say anything extra if you want to, and start to share your screen whenever you like. Thank you so much, everyone.
So thank you, Ellie, for such a splendid introduction. So I'll just begin. I welcome you all to my presentation on review of mineral accretion technology and electrotherapy as tools for marine habitat restoration. As Ellie pointed out, I have recently completed my graduation, and this was my topic for literature review on which this presentation is based. This literature review was primarily completed under the guidance of my supervisor, Dr. Srireka PR, assistant professor at the Cochin University of Science and Technology. Throughout the course of this talk, if I go too fast or there is some error, you do not understand something, please feel free to interrupt me at any moment. If you have any questions, you can ask me after the talk ends. To begin with, I would like to talk a bit about my inspiration for taking up this topic for my literature review. So basically, in 2017, I joined a group of marine enthusiasts named Marine Life of Mumbai. And my seniors at Marine Life of Mumbai introduced me to this species of corals. And this is the first time I was actually seeing a coral. And to my surprise, this was based exactly in my neighborhood in Mumbai, and I was quite surprised by the fact that we have hard corals living in the polluted waters of Mumbai, because whenever we imagine a coral reef, it is usually those blue tropical pristine waters, but here it is right at the boundary of the anthropogenic stressors. And this is a very big pilgrimage site in Mumbai named Haji Ali, and here we have hard corals, which are of course protected by the law. Now, what fascinated me more was that it used to change colors every time I observed it. So this was taken in April, and we can see the coral colony in its full splendor with the bright red color. Coming during May, the temperatures increase in India. So during the summers, it kind of turns whitish. In June, that is the monsoon period, when we have rainfall, it completely turns into a whitish white color. And again, it starts regaining its color in November, December are the post-monsoon months when the temperature is quite low, or not quite low, it's around the 20 to 30 degrees Celsius, which is ideal for corals. And we start watching these corals regaining color. So this is the type of resilience that this coral species, it is called a false pillow coral, Pseudosiderastria tyami. This is the type of resilience that it shows. And why does this occur? So primarily the theory says that coral, although it is an animal, it lives along with a plant. So there are tiny plants called zooxanthella that live inside a coral animal, a coral polyp. Now, as we know, plants photosynthesize. They prepare their food using sunlight, water, and carbon dioxide. So inside these corals, these plants, the plants named zooxanthellae, will photosynthesize and create food for the corals to survive. In return, coral offers a lot of services. First of all, these plants get a comfortable place to live inside the coral. Coral will give them carbon dioxide that it produces via respiration, and also the nutrients that are required to carry out photosynthesis. This type of relationship is seen in a lot of different organisms throughout the natural world. It is called mutualism, in which both the partners, in which basically organisms live in under a partnership, and both the partners are benefited. This is what fascinated me, has been fascinating me since 2017. And back in February, I posted a random query on a group on Facebook that I'm looking out for topics to do my dissertation on. And Mr. Chad Scott, the CEO of Conservation Diver, suggested me that I explore the potential of mineral accretion technology, which can be used to artificially increase the resistance and resilience in coral reefs. So I immediately jumped upon that technology, and yeah, this is the product. This presentation is a product of that literature review. So this was my motive behind this review. Mineral accretion technology is also known as Biorock. It is an extremely innovative technology that can be applied for restoring important marine habitats such as coral reefs, salt marshes, seagrasses, as well as other advantages like increasing fisheries potential, tourism, and in coastal protection. Now, this review was an attempt to decipher the efficacy of using this technology in ocean sciences, comprehending its various uses, limitations, and filling up the gaps in past literature, as all literature reviews do. Through this presentation, I will try to decipher the answers for these four major questions. First of all, what plausible mechanisms are involved in the working of this technology? Secondly, what is the efficacy of this technology in restoring important marine habitats, as I said? Thirdly, how can mineral accretion technology be applied in fields other than marine habitat restoration, which has been highly underexplored right this topic? And last, what can we expect from this technology in the future, and how will it evolve? Methodology used for this review was straightforward. Literature was obtained from peer-reviewed articles, but this topic also needed searching the gray literature, which includes unpublished articles, theses, websites, and personal communications, because many of the literature that is available for this technology has not seen the light of peer-reviewed journals. And I'll come on to that later in my talk. After that, I just search on Google Scholar using these keywords, and one for each of my chapter, like restoring coral reefs using Biorock, then oyster, salt marshes, etc. And the results obtained were quite large, but actually studies relevant to my topic were very few out of them. So I had to filter them accordingly. And similar searches were made for generalized topics like valuation of ecosystem services, threats to marine ecosystem, climate change, sea level rise, tourism, etc. And finally, all the literature that was obtained was organized section and year wise using the software Mendeley Desktop. Any idea how I can remove this panel on the top?
The panel you can move around, or you can just make it smaller. But it's only you that can see it, so it's not blocking the view for us.
Okay, but it is obscuring my view just a moment.
Okay, so you should just, you should just be able to drag it to the side. Okay. Or if you're not in full screen mode, then it should not overlap with your presentation. Okay,
just a moment. Sorry for this. All right. So introduction, like what I feel is that our planet Earth is a misnomer, because oceans cover 71% of the Earth's surface actually. So I feel that our planet should have been named planet Oceanus. But despite of covering such a large surface area, this ecosystem is under threat. Our marine ecosystems face a lot of threats, which include climate change and climate extremes in the long term, physical and chemical alterations of seawater, marine pollution, of course, which includes domestic as well as industrial waste, over exploitation of resources, and lastly, habitat destruction and degradation. Now, these factors bring about a lot of threats to the marine ecosystems as well as the organisms that reside in these ecosystems, such as changes in the organisms' genes and physiology, range shifts, loss of valuable ecosystem services, and endangering and extinction of marine flora and fauna. These threats can be mitigated to some extent using passive using passive measures such as declaring a site as MPA or reducing the stresses that stress this ecosystem. But they have been, some of them have been unsuccessful and they are not completely effective. So I suggest that active restoration is the need of the hour. It can be defined as the reintroduction or augmentation of a particular species to protect it, but often at a higher cost. So mineral accretion technology is an active restoration measure. It is defined as the third generation of artificial reefs technology. Now, here's an interesting thing. Even Charles Darwin was aware about the artificial reef, of constructing artificial reef. He once saw a coral fragment, a broken coral fragment, which had no hopes of surviving. What he did is he attached the coral fragment to a bamboo pole and suspended it into the seawater. Miraculously, that coral fragment ended up living. So what he did was he transplanted a coral fragment into the onto a bamboo pole and inserted it into a seawater. That is the second generation. And even before that, people since thousands of years have been inserting artificial habitats such as anything, a pole, a wood pole or a rock inside the seas to attract fishes. That is called FADs, fish aggregating devices. So FADs were the first generation artificial reefs, because of course coral larvae would kind of get settled onto those structures. Now, principle behind this technology is quite simple. Our seawater is not pure water. It is a complex mixture of cations and anions, and an increased pH of water leads to the precipitation of these compounds on a given substrate. It was invented by this great man, Professor Dr. Wolf Hilberts, in 1976. He was basically an architect. So his intent was not to actually put this technology to work in marine habitat restoration, but to actually extract solar generated building material from the sea. So he thought, yeah, okay, there is a lot of CO2 outflux coming from cement manufacturing. Why not we there's a lot of calcium available in the oceans. Why not we generate calcium out of the oceans and then we apply it to building materials using solar energy. So he originally named it as mineral accretion technology, secret or cement. And then in 1987, he was invited to Jamaica by Dr. Thomas Guro, who is a marine biologist. And they started applying this technique for constructing artificial reefs. They called it Biorock and trademarked it. And they also founded an organization named Global Coral Reef Alliance, which is still active in and provides services for the application of this technology around the globe. Now, this process is a simple 5-step process. There's nothing complicated in it, which involves the installation of cathode and anode into the seawater, first of all. Secondly, it involves the application of a steady pulse or intermittent direct current. It is important to note down that only direct current will work and not the alternating current. It will immediately fail if we provide alternating current across the 2 electrodes, and current density should be really small, just around few amperes per meter square. But different researchers have used a different current density. Then mineral starts getting accreted on the cathode. Accreted means starts building up slowly. There is a recruitment of aquatic organisms on or near the cathode. And there is a creation of highly alkaline conditions and high electron density for higher growth rates of organisms on the cathode. So we'll look at a little bit of chemistry here. I won't delve too deep into the chemistry as it would complicate things. But of course, there are 2 electrodes here, anode, which is positively charged, and cathode, that is negatively charged. The things to look out for here are that at the seawater, there's seawater in between the 2 electrodes. So we have the calcium ions here, and calcium ions will react with the bicarbonate ions, HCO3, and form calcium carbonate. This calcium carbonate will get accreted on the cathode. At the same time, seawater has magnesium ions, which will get reacted with the hydroxyl ions and form magnesium hydroxide on the cathode, along with silicon oxide and sodium chloride, but we are mostly dealing with calcium carbonate over here. Also, in electrochemistry, cathode is the reducing end of a cell. So water, when it obtains two electrons, because that is reduction, obtaining an electron by a chemical species, it gets reduced to hydroxyl ions and liberates hydrogen gas. So that is how we know the technology our plants are working. Once we start supplying uh, electricity across the cathode, we should see hydrogen bubbles that being liberated out of it. And at the same time, anode is the oxidizing end, right? So water gets oxidized and releases two electrons as well as two hydrogen protons. These protons make the anode acidic, and since over here hydroxyl ions are being liberated, they make the cathode alkaline. I hope this is clear. Another thing that we need to look out for here is this two protons can combine with chloride ions and form hydrochloric acid, which dissolves the calcium carbonate. That is why cathode should be placed at a distance away from, the anode should be placed at a distance away from cathode. So there are three components that are involved in the working of this technology. That is the cathode, anode, and the stable power supply. So cathode is made up of a conducting metal that is usually iron or steel, and the shape is flexible. It depends on the practitioner who needs to use this technology. It could be cylindrical, sheet like triangular, etc. And anode can be made up of any shape. Okay, because that is not the electrode that we'll be transplanting our organisms on, but it is made up of graphite, lead, titanium, and alloy of platinum or titanium or metals coated with their oxide. Basically anything that is a good electron donor to the cathode. But titanium anodes work the best according to Zamani, but at the same time they are very expensive. And the shape can be of any shape, as I said. And lastly, the most important aspect, that is the power source, can be used. There is land-based. Ellie, I'm getting a message that my internet connection is unstable. Am I still audible?
Yes, there was a second before where it dropped out, but not enough that we couldn't get the sentence or anything. But you're good now, at least from my position here.
Yes, we can still hear you.
Okay, perfect. Okay, perfect. Thank you. Okay, so there are three power sources. There is land-based power, there is land-based battery power, and solar energy. The best one and the most reliable one is using land-based power, the AC power that we get. So it should be paired with a step-down transformer as well as an AC to DC transformer, because as I said, direct current is needed for the working of this technology. But also it questions the sustainability of this technology, because a lot of fossil fuels go into producing land-based power. Of course, there are other means, but yeah, still fossil fuels and coal is needed sometimes. Then comes land-based batteries, which already provide DC power. So we just need to pair it with a step-down transformer and a stabilizer to achieve the amount of voltage that we need. And lastly, the most ecologically sustainable is the solar panels. If you use solar panels, the power can be stored in a battery or they can be supplied wires. But again, this hampers our project sometimes, because solar panels won't work when there is less sunlight during cloudy days, at nights, or sometimes throughout the seasons. Suppose in India we have a monsoon season when we get very less amount of sunlight, solar radiation. Also, it won't work outside the tropical region in the temperate. Here are the various designs that was used. This one is the earliest by Hilberts in 1977. This is by Sabater and Yap, quite recent in 2004. We can see coral nubbins being planted on the cathode. This wire mesh is the cathode, and these graphite bars are the anode. And here we can see there are wire connections there. And this one is a recent one by Jompa in 2013. This was used in Indonesia, where they have attached a floating solar cell on the surface of water, which powers the cathode and the anode directly. Now, assumptions for its working. Why does it work? The main reason for its working is the alkaline conditions around the cathode. As stored in this reaction, there is high amount of high concentration of hydroxyl ions that are being generated around the cathode, and this carbon dioxide actively,
Vignesh,
which
maybe we'll
induces
just
the
pause.
precipitation of aragonite. Aragonite is a form of calcium carbonate that the, sorry. Hello.
There was just a moment there where you were slowing down a little bit. It seems to have fixed itself up though. Although while we've paused,
Okay.
did you want to do a question now or a question at the end? Someone's just sent one through to the group chat. Which is
question at the end.
Sure. Okay, we'll do it at the end. I think your audio is good again now. I just wanted to let you know it was a little bit laggy, but okay, go ahead.
Where was I lagging? I can repeat always.
It wasn't significant. It was just sort of getting a little bit down. But I think I didn't feel like I missed anything significant. So go ahead.
All right. So coming on to the assumptions for its working. So corals maintain a naturally alkaline environment between the calicoblastic cell epithelium. These are specialized cells that are present in the ectoderm of a coral polyp and the substratum to which the coral is attached. By removing carbon dioxide actively, and this induces the precipitation of aragon, which is used by the corals to build their exoskeleton, their outer skeleton. Now, having a high density of hydroxyl ions around it, cathode causes a similar deposition of orthorhombic aragonite crystals artificially, and this increases the availability of calcium carbonate for calcareous skeleton building organisms. Secondly, there is supersaturation of calcium and carbonate ions in the vicinity of the cathode, and this actually magnifies the rate of buildup of the calcareous coral skeleton. So researchers previously thought that only calcareous organisms, means organisms that build their exoskeletons using calcium, are getting benefited by this technology. But it was later observed that non-calcareous organisms such as seagrasses, salt marshes are also getting benefited. Then came another two assumptions. Yeah, so higher availability of free electrons around the cathode. So higher availability of electrons means higher efficiency in cation uptake across the membranes, because electrons do store a lot of potential energy, and then when they are passed from one electron acceptor to other, they release some of this energy, which kind of motivates the the proton pumps to take in cation and take in cations inside the membrane, and less oxidation of macromolecules. So in our body, we have a lot of macromolecules like carbohydrates, produce free electrons for working of the electron transport chains, and these factors leads to a higher growth. So we don't need to oxidize these macromolecules if we have free electrons already available, and these macromolecules can then be used by the organisms to build up to increase their growth rate or to reproduce quickly. Then there is increased production of ATP. A study conducted by Cheng et al. In 1982 showed that it was conducted in artificial environment on a rat skin tissue, showed that ATP is the energy currency, as we all know, of our body, of a cell. ATP levels increased by 3 to 5 times at currents when stimulated at currents between 50 microamps to 1000 microamps. And this increase in biochemical energy can stimulate growth, reproduction, healing, and resistance to unfavorable environmental conditions. And lastly, electrotropism, but this point is related more to an increase in larval settlement that is observed in some of these structures. So what Dr. Guru predicted is that embryonic and larval forms of marine animals and plants possess a head to tail or a positive and negative polarity. These larval forms get aligned according to their polarity and are attracted towards the electric field, leading to an enhanced larval settlement on these Biorock structures. But again, this point has been challenged by a few researchers. So coming on to my first topic, that is coral reef restoration. These are the important studies that I found in my literature when searching for my literature. So it can be classified as both physical restoration and substrate enhancement. Corals grown on Biorock have higher growth rate, branching, girth, settlement, healing, resistance to stress, etc. but it is important that we have periodic live and dead phases, means phases with electricity and without electricity, because Schumacher and Schillac observed that larval settlement was taking place only when current approaches zero. So this goes against the assumption of Dr. Guru, as I previously mentioned. And this technology has been used to revive reefs that have been mechanically damaged, due to ship strikes or some natural calamity. Another influential paper by Dr. Guru in 2004 explored the physiological alterations in corals that are that grow on Biorock. So they have higher zooxanthellae density, mitotic indices, and fluorescence. Higher fluorescence means better rate of photosynthesis, as well as lower chlorophyll density per zooxanthellae. It has also been used to increase the growth rate of cold water corals, Lophelia pertusa in Sweden. It has shown promise. And in my country, India, Biorock project is being undertaken by the Zoological Survey of India in the Gulf of Cochin, Gujarat. Internationally, it is usually applied in the countries of the Asia Pacific, that is the Coral Triangle, such as Indonesia and Philippines. So yeah, this technology, of course, is highly successful, and it can revolutionize the field of reef restoration, right? But here's where the things get a bit complicated, and some researchers have also challenged the working of this technology. So an in situ study conducted on Acropora pulchra and Acropora youngae, both of them are hard corals, showed high mortality and lower growth rates, lower chlorophyll concentration, zooxanthellae density, and relative electron transport rate than the control corals. So this kind of challenges the success of mineral accretion technology. But Dr. Guru was not so amicable with these results. He refuted this claim, and in a later publication in 2014, he claimed that a lower growth rate was the result of selective herbivory by a male parrotfish, which was not mentioned in the publication by Borel. Later on, it was found that over the long run, cathode becomes oversaturated with the accreted mineral, and this leads to a reduced interaction between the two electrons until the accretion completely stops on the cathode. And in Benedetti in 2011 observed 5 times slower settlement of the planula larvae of Corallium rubrum. This is a soft coral on electroaccreted calcium carbonate with polarization. With polarization means under electrical stimulation, then control. For this study, control was just a marble, a piece of marble, and accredited calcium carbonate without polarization, means without electrical stimulation. Now I know that
Vighnesh?
this graph looks a bit scary, but trust me. Yeah.
Sorry, your last comment. Someone just suggested, it's going pretty well, but there are a couple of points here and there where you cut out for just a second. Maybe you can try maintaining the share screen, but turning off your video might be a good idea.
All right.
And then, but otherwise it's not too significant, so don't worry. And yeah, go ahead. This is great.
Okay. I hope I'm audible now.
Perfect.
Okay, so what I was trying to say in the last point was that basically larval settlement on mineral accretion technology plants with electrical stimulation was reduced as compared to when it was not under electrical stimulation. So electricity kind of repulsed the larval settlement in this study by Benedetti. Now I know this looks a bit scary, but this is a very cool result obtained by Sabater and Yap. What they basically realized is that corals being grown on Biorock projects or mineral accretion projects had alterations in the colony morphology. Usually how it should be is that mean corallite diameter should be the least at the tip, because that is the actively growing part of the coral colony. Then it should gradually increase in the middle section and maximum at the base. But when we look at the treated section, the coral nubbins that were actually stimulated by electrical current, we see that of course it is very low at the tip, it increased in the middle section, but again it drastically reduced in the end. So we get a bell-shaped curve like this, and that should not be the case. It should be straight like all these graphs. And the reason they predicted for this is that calcium carbonate gets accreted directly on the cathode surface. So correlates at the coral polyps at the base have a lot of interaction with the calcium ions, and usually these coral polyps, they spend energy for the uptake of these calcium ions. Since calcium ions are in high quantity and available for free, they can rechannel the excess energy for reproduction, and that is exactly what they did. They kind of use the excess energy to reproduce, to bud, using budding. And this was what was found, that mean correlate diameter kind of increased in the middle section and then decreased. So this discussion as to why this is relevant in this day and age. First of all, we live in an age of global warming, which is drastically increasing the sea surface temperatures. And calcium carbonate deposition actually increases when the seas when the temperatures rise. But at the same time, high temperatures leads to coral bleaching, which leads to massive coral die-off. But this technology has shown that corals grown on Biorock plants show a higher resistance towards coral bleaching. So yes, they can be used for fighting our fight against global warming and higher sea temperatures. Secondly, corals grown on Biorock have shown increased resistance towards coral bleaching. And third, alkaline conditions created around the cathode protect corals against the effects of ocean acidification, because a lower pH, an acidic pH, can dissolve calcium carbonate and lead to coral mortality. So it protects the corals against these. And this technology can be used to restore coral reefs in various parts of India as well, because we do have some patchy coral reefs as well as atolls and other types of coral reefs. Now coming on to my second topic, which is oyster reef restoration. Studies conducted on them have been very meager. So oysters grown under electrical stimulation have a higher growth rate and lower mortality even in winters. So winters are a harsh time for oyster to survive, especially in the middle latitudes, because colder temperatures, they lead to the dissolution of calcium carbonate, through which these oysters prepare their shells. That is why sometimes even 99% mortality is observed in natural populations. But that is not the case with when they are grown on Biorock. We can see here a researcher is artificially transplanting oysters onto the cathode. And this can be attributed to, the success can be attributed to high deposition of calcium carbonate around the cathode, higher production of ATP, and formation of a macromolecular shell matrix, which increases the efficiency of calcium carbonate deposition, and it makes the shell stronger. So oyster reef restoration methods face a lot of challenges, the traditional methods, and they are hampered by non-availability of culture materials, perils of introducing too many foreign materials, high mortality in polluted waters, and in extreme climate events such as winters or snowfall. But mineral accretion technology can be used to restore natural populations even in the most polluted estuaries and bays where they were once abundant. And this is very eminent because oysters are kind of natural filters of the seawater. They reduce the turbidity of water, of a water column, and make it clear, which also increases the productivity of the water in the long term. Then salt marshes, of course, Biorock technology has the capacity to restore salt marshes even in the most polluted and harsh environments. For this, I would like to cite Servino et al., a study that I really liked. They transplanted the salt marshes in America's most densely populated city, that is New York, between the in the East River, between the New York Central Park and landfill, an unused landfill, which used to leach out a lot of pollutants like heavy metals and other organics like polychlorinated biphenyls and polyaromatic hydrocarbons into the water. And they wanted to test whether these salt marshes, Spartina to be more precise, survived in such harsh conditions if they used this mineral accretion technology and stimulated it with electricity. And yeah, the results were quite favorable, as we can see here. These diamond-shaped ones are the ones that were stimulated by very high, not very high, but high electrical, sorry for that, was stimulated by high current density, and they showed a very exponential increase in height, as we can see here, as the days progressed. While those that were not stimulated at all, the currents are the circles here. So we can see their growth rate has been relatively low. So what I suggest is that this technology, that is Biorock, can be used to artificially increase the growth rates of C4 plants. In photosynthesis, there are in the plant kingdom, there are two types of photosynthetic plants. There are C3 and C4. Some publications have stated that in the future, the conditions of C4 plants will be paralyzed because high CO2 in the atmosphere favors C3 plants. So C3 plants will kind of outcompete the C4 plants in the future. And many of the genera of Spartina and Phragmites are C4 plants. So we can use this technology to artificially increase the growth rates of C4 plants in mixed colonies to make sure that they survive in the future. But there should be no introduction of exotic species. Now, this study used only one parameter to assess the success, that is the plant height. But there should be other parameters to test the growth and health of plants, such as surface area, leaf and shoot number of stem clusters, etc., and the photosynthetic capacity as a physiological marker. Then there is a need to perform experiments in salt marshes plants other than grasses, because in India, 50% of salt marsh species are succulents, and there is need to check the efficacy of a restoration project with a larger spatial scale. Now, seagrasses, again, just like salt marshes, they are very important ecosystems. They store a lot of carbon as blue carbon and sequester a lot of carbon as well. They are also called as the lungs of the sea. So seagrasses grown in situ under low electrical stimulation have shown a very high growth rate, even in a hostile environment, as noted by this publication by Vaxarella in Italy. This was conducted in Italy. What they did is basically carried out the seagrass restoration project using Biorock at 4-meter depth at a site named Giovinazzo, and at 8 meters or depth or more at a site named Torre Guaceto. Okay, so this one was quite favorable for the growth of seagrasses, this site, but this one was macroalgae dominated site. So what they observed was that at this site they stimulated the seagrasses with some high electrical density, and that led to the high rate of accretion of calcium carbonate, and eventually it ended up smothering and killing the seagrasses. But over here the current density was quite low, and we can see that the seagrasses survived well and their growth rate was also nice. At the same time, they also saw that the density of organisms around that depend on seagrasses increased around the seagrasses, which includes annelids, crustaceans, mollusks, and echinoderms. So thriving seagrass ecosystems can be revived using the Biorock technology, but there is a need to standardize this technology for multiple species of seagrasses, and there is a need to check the feasibility of applying this technology to create multi-species patches as well, because a genetically diverse community is better suited at handling stress and surviving. And apart from the growth rate and survival, flowering capacity and the period of flowering of the transplanted seagrasses should be taken as an essential parameter for health. This is a very intriguing topic, that is its impact on fisheries and aquaculture. So researchers at the Gondolin Research Institute of Mariculture in Bali released 3 species of grouper hatchlings into the Biorock structures, and they continuously monitored these hatchlings. They tried to attempt restocking using fish restocking using Biorock technology. Now what they observed was that these fishes, they grew steadily for a period of about 3 years around the reefs, and then they migrated. They suddenly disappeared to deeper water. So this experiment was successful, and a publication by Jompa in Indonesia observed the fish populations around the Biorock structures as well. And there they observed a clear-cut increase in coral reef density, evenness, and diversity around the Biorock structures. Another innovative method is called post-larval capture and culture, which is developed by a French company named Accretion. What this involves is basically the young forms, the baby fishes, they experience a lot of mortality, around 90 to 95% mortality, sometimes due to predation or some other factors. So we can attract them using light and then trap them. After trapping them, we can artificially grow them in aquaculture ponds, and then when they reach a considerable size of survival where mortality will be negligible, they can be again reintroduced into the Biorock structures. So through this, we bypass the predators, introduce a multi-trophic fish structure, and reintroduce the wild genes back into the nature, hence preventing genetic pollution. So why does this technology have a lot of potential in fisheries and mariculture in the future? Because first of all, these reefs can be modified and shaped as per the needs of the species that is being grown. So suppose we want to grow lobsters, there need to be a lot of holes inside the Biorock structures, and that can be made possible. That is not possible in natural reefs. Of course, there are hiding places in natural reefs as well, but this has been artificially manufactured to grow a particular species. Secondly, only source of energy supplied is electrical energy, and there is no need to supply extra feed. Thirdly, it gives a pluriculture as opposed to a monoculture practiced in traditional mariculture. And lastly, in cold water areas where coral reefs cannot survive, we can use oyster reefs to create a Biorock for mariculture. However, Biorock technology has a negative impact on elasmobranchs, means sharks, skates, rays, basically fishes that do not have bones. Because these organisms have minute electroreceptors called ampullae of Lorenzini. And it has been predicted that this technology, since it uses electricity, kind of screws up with their electroreception, and we see altered behaviors such as avoidance of the structures completely, reduced feeding. So this might have negative implications because sharks are the apex predators of the marine ecosystems, and apex predators are always important for the well-being of a particular ecosystem. So proper assessments are required before setting up Biorock projects. Another innovative method, another innovative use of Biorock technology that I noticed is using it for preventing beach erosion. Now, due to the sea level rise, beach erosion has become common throughout the globe. So Biorock has shown promise to reduce beach erosion, or rather build up new beaches as well. So it has been categorized as a soft option of beach erosion, and the project was carried out at Pulau Ganga in Indonesia to restore an eroded beach using Biorock anti-wave structures. So in 2015, that beach, the Pulau Ganga beach, majority of the beach had eroded and formed a 1.36 meter high erosion cliff along the shore. As you can see here, there is a concavity due to erosion like this. But in 2017, there is no concavity here, and the beach has completely recovered. Now, why did this happen? Because in 2016, 48 Biorock anti-wave structures in a staggered design were built and deployed on a seagrass bed adjoining the sea. Now, these tiny white dots here that you can see, these are the Biorock structures that were deployed. And the result was that approximately 80% of the beach grew back within 3 months of the deployment. And their success is quite evident. If we look at the southern portion of this beach, there are no Biorock anti-wave structures here, and the erosion is evident by the concavity. And over the course of a year, the beach height increased by a meter and width by more than 15 meters over a 200 meters length. So why is this successful? What mechanisms are involved in preventing beach erosion? First of all, Biorock structures can be installed along the beach to successfully prevent beach erosion. And these frameworks dissipate the wave energy using mechanisms of internal refraction, diffraction, and frictional resistance. the traditional methods of preventing coastal erosion, such as deploying tetrapods or building seawards, what they do is they basically reflect the wave action, and in that process get kind of eroded themselves. So that is not a viable solution for the long run. But these are, secondly, wave energy gets further dissipated by crustose coralline algae, soft and hard corals, oysters, etc. The organisms that get settled on these structures naturally. And lastly, they are more resistant to destruction by natural calamities like storms and hurricanes as compared to other structures. So a publication by Wells in 2010 showed that uh, deployed Biorock structures in the Caribbean resisted at least 2 hurricanes, uh, like that. So yeah, it is successful and more resistant to destruction. However, the projects require a continuous supply of electricity and will fail if the power is cut off. And they have indeed seen failures in Maldives because they cut the power source after a few months. Because what happens is when you cut the power source, the cathode will start eroding without the support of the anode, and eventually the projects will fail. They will dissipate. And lastly, the influence of tourism. So coral reef tourism is a multi-billion-dollar industry and has been increasing over the past few years. But over tourism can cause a considerable stress to the local reefs. But Biorock reefs can help ameliorate some of the pressures that are being put on natural reefs. First of all, they increase the number of tourists visiting a particular area. They increase the fish diversity around them, as I pointed out in my previous slides. And that helps attract a lot of tourists in the form of snorkelers and divers. A study conducted at Koh Tao by a researcher at conservation diver named Nichols indicated that tourism can be augmented if these reefs are placed in close proximity to a natural reef. So tourists can enjoy both the Biorock reefs as well as the natural reefs. This study also pointed out that shipwrecks around Koh Tao attracted maximum number of tourists, and that is true. Globally, shipwrecks attract the maximum tourists, so they can be theoretically converted to Biorock reefs because ships are made up of iron metal, but that would require a substantial amount of energy, which is unfeasible if supplied from the land because of the resistance offered by the wires that carry electricity. And this technology can be propagated as an example of responsible and sustainable tourism. So here are some challenges and difficulties that I observed that need to be taken into consideration. First of all, there is lack of standardization, and standardization is extremely difficult as well. But we need to decide upon a fixed set of measurements to determine coral growth for each growth form. There are different types of coral growth forms, like branching, digitate, crustose, etc. A lot of them, folios as well. So what we need to do is we need to fix a set fixed set of measurements to determine coral growth, because each researcher has used their own proxy for this thing, for coral growth. Growth is not well defined. Some of them have used girth, some of them have used longitudinal growth, some of them have decided the growth of corals depending on the branches or the length of the branches. So all these things end up making the data a bit precarious. At the same time, a success of restoration project depends on how well the restored habitat achieves its original state, and that is possible only through the reproduction of the organisms that have been introduced or transplanted. So time of first spawning needs to be considered as a parameter for health, for testing the success of a restoration, and I found no studies checking the time of first spawning. And secondly, there are of course too many proxies, as I said, such as chlorophyll A concentration, rate of photosynthesis, chloral fluorescence, etc. And that is the reason there needs to be an increased coordination between Biorock practitioners around the globe. So I assume that there is a robust community of coral reef restoration practitioners, and a committee can be set up, and the standardization of the parameters to check coral growth and health can be set up. Thirdly, there is a need, need for expertise, because people working in this field need to be trained, not just theoretically but practically as well. And hiring professional experts can increase the expenditure of a project. That is why many of these projects are voluntary driven. So the volunteers should be trained perfectly. Then there are lack of long-term studies. You know that the growth of coral reefs takes place over a long period of time, but most published studies report a duration of less than a year, and this gives a false impression of growth accretion in acceleration in organisms. uh, because of course they will show a higher growth rate under electrical stimulation initially, but that might be not be the case after 2 or 3 years, and hence it fails to provide us a clear image of whether this technology is indeed a success in restoring important marine habitats. Secondly, there is a severe scarcity in literature. This technology was invented over the past 25 years ago. The patent was obtained in 1996, but there have been very few publications over this time period, and this might be due to the fact that patented technologies like this come with a patent fee, and not everyone can afford this hefty fee. But the patent has expired now, so the technology and the raw material itself is quite cheap, and practitioners can start using it. Also, I noticed some biased publishing. Studies published on coral restoration is in general are biased towards those showcasing only the successes. So failed projects or projects that did not get the desired results do not get published. They get obscured, but it is important to publish that data as well, so a future researcher can read the literature and try to solve the problems that were faced by the studies that were unsuccessful. At the same time, publications on scleractinian corals, that is the hard corals, heavily outnumber other taxa such as oysters, salt marshes, and seagrasses, and there needs to be more transparency in publications. There are, of course, logistical problems. We are dealing with large structures. Also, we are dealing with natural ecosystems such as the sea. So there can be natural causes such as storms, rough seas, earthquakes, tsunamis, etc., that can spoil the project, as well as some man-made mechanical damages like dredging, sea strike, wires getting cut by a propeller, etc. And there needs to be lack of constant surveillance. There needs to be constant surveillance because a lack of surveillance can hamper the results. At the same time, there should be constant maintenance as well. We need to remove the fouling organisms such as barnacles or macroalgae that settle and compete with the organism that has been intended to that we have intend to restore. Then we need to fix a carbon budget for this technology, and I think this is impossible to do, but still attempts can be made. Usually, when corals build their skeleton, they end up liberating carbon dioxide into the water. So calcium ions plus bicarbonate leads to the deposition of calcium carbonate and give out carbon dioxide. Now, it would be ironical if this technology is a source of carbon dioxide, because here we are talking about saving our futures with technologies like this. But of course, the amount of CO2 released into the water and eventually into the atmosphere because of that flux is quite small as compared to the industrial emissions. However, when seawater is starved of calcium, it will try to obtain calcium from other sources, such as calcium silicate minerals that are our sand is mostly calcium silicate. So what will happen is calcium silicate minerals plus water, when water reacts with calcium silicate minerals, it uses carbon dioxide to form this mineral calcium bicarbonates along with silicon oxides. So designing a carbon budget for individual Biorock projects is indeed a daunting task, and we need to consider the ecosystem being restored as well as the sources of energy. Because yeah, if the source of energy is the land-based power plants, which comes from burning the fossil fuels, of course it is unsustainable and a source of carbon dioxide. Then there are high costs that are incurred in these projects. So there is a cost. It has been estimated by Guru and Hilberts into a publication by in 2005 that the cost of protecting shorelines equals to approximately $50,000 to $100,000 per kilometer. But again, I don't think this is a very big sum. Also, they predicted that cost of restoring one-tenth of the world's most important coral reefs would amount to around $300 billion. Also, critics state that this technology is not cost effective and requires constant maintenance. Here are some future directions that I suggest. First of all, we need to determine the current status of Biorock projects. So I suggest an online questionnaire based survey that can be carried out using the power of digital and social media. A questionnaire can be prepared with questions pertaining to various aspects such as the type of cathode used, anode used, the budget available, location, the type of species that are being transplanted, etc. And they can be sent to various practitioners all around the globe. And based on the answers and the data that we obtain, a database can be built up, or an online database, and that can be freely accessible. So it will help practitioners around the globe. Secondly, using fluorescence as a reliable proxy for coral health. This can be done using the pulse amplitude fluorometry. This is a completely non-invasive technique of determining fluorescence. And why fluorescence? Because fluorescence is a diagnostic biomarker for determining the rate of photosynthesis in the zooxanthellae. So if the rate of photosynthesis is high, this means there is high amount of carbohydrates being produced, and this is the food for corals. So of course, if the corals get more food, they will grow quickly. And there is also a need to detect correlation between coral fluorescence and applied electric field. That is, if we increase the electric field, will the coral fluorescence increase as well or not? If it does, then it of course it is a reliable proxy for coral health. Then assessing the holobionts. So so far I've talked only about association of corals with the zooxanthellae, but coral microbiome, that is microorganisms living inside the corals, consist of a lot of bacteria and fungal species as well, which make mammoth contributions to coral health. There have been recent studies that point out that these bacteria help corals to fight against coral bleaching. They act as probiotics actually. And there is need for more research on whether electrical stimulation can change the composition of this coral microbiome, and this can be achieved via culture dependent or independent methods. Then how can we do this? So we need to incorporate the omics technology. We live in the age of omics technology. Terms like genomics, epigenomics, transcriptomics are household terms these days in molecular biology. So we need to start exploring the potential of molecular biomarkers in Biorock restoration efforts. So I'll explain this with an example. Now, theory says that an enzyme named carbonic anhydrase is used by the coral polyp to build up its carbonate skeleton. So what we can do is we can look at the genes of the coral polyps that are being grown on Biorock structures under electrical stimulation, and then we need to determine transcriptomically. It means need to look at their mRNA to determine whether there is some hyperactivity of gene that is responsible for synthesizing this protein named carbonic anhydrase. Or we can also see some epigenetic markers, whether growth inhibiting factors are being methylated by the coral. So there is a lot of potential in applying omics technology in coral restoration. Then of course we need to determine the optimal growth condition, because there is a lot of ambiguity in relation between electrical density and growth rate of coral colonies available in the past literature. So current densities need to be optimized for each species or ecotype, and this might be challenging because different species behave differently in varied habitats. Then there needs to be an increase in the spatial scale of these projects. So most of the studies that I described so far have been experimental in nature, but we know that marine ecosystems are large ecosystems that stretch for thousands or tens of thousands or millions of kilometers. So is this really feasible for restoring such large ecosystems? And for that we need to take into consideration some points, such as, first of all, its cost, because it costs on a per meter square basis. Then we need to ensure that there is a constant electrical supply and maintenance, high costs of the solar panels. Again, maintaining solar panels is a difficult task. And yeah, that is why we need to check the feasibility of these projects. And in the future, we run into the risk of building monospecies reefs, because there is a tendency to transplant only the fast growing species, such as the branching coral acropora, because the researchers obtain fast results when they do this. But that is not ideal, and we run into the devil that we wish to abolish in the first stage, because a monospecies reef will have a low genetic diversity, and it might not be able to tolerate stresses and might bleach quickly. So it is essential to introduce diverse species of corals on a Biorock structures, and I assume most of the practitioners around the globe are doing that. And lastly, we need to combine uh, Biorock projects for multiple ecosystems, because no ecosystem can exist in seclusion. So what we can do is we can build multiple ecosystems side by side. So there is a coral reef. Besides that, there is a seagrass, thriving seagrass community that is growing. This will also benefit the fish species that are growing. So because some fish species have life stages such that they kind of the adult stages live in the coral reefs, but spawning and hatching takes place in the seagrasses or in salt marshes. There are many species like that. So a Biorock mega project consisting of a large number of deployed Biorock structures can be designed in the future to incorporate multiple ecosystems within an area. To conclude, electrotherapy and mineral accretion seems to be a revolutionary tool, of course, for the recovery of degraded habitats. It will augment local economies and help spreading awareness about the importance of marine habitats. Fishermen can turn into farmers using this technology. It will boost tourism and hence augment local economies. However, it is still in the juvenile stage, and several questions need to be answered, as I pointed out in my previous slides. And it is by no means a substitute for marine conservation or an excuse for overexploitation. So I guess what I suggest is that the traditional forms of marine conservation, such as MPA management, should not be stopped, and this technology can be used as a partner for those methods. At the same time, there is a negative attitude sometimes among people that okay, we can exploit the corals or fishes now, and then we can invest in this technology in the future and regrow the ecosystems. That is a wrong attitude, and it should not be practiced. So the final question, will this technology be the answer to our global crisis of marine habitat destruction and degradation? Only time will tell as the technology progresses. These are the important references that I have used in this presentation. So I thank you all for attending this talk and spending your precious time on a Sunday. I would like to especially thank my guide, Dr. Srireka, PR, and last but not the least, Mr. Chad Scott, the CEO of Conservation Diver, because he helped a complete stranger like me and answered each and every query of mine with the utmost detail and patience. So I hope it was not too boring. Thank you. Thank you very much.
Thank you so much. That was really, really interesting. I think I can speak for all of us when I said that was really, really, when I say that's really great, your presentation. There's been a series of questions that have been coming through while you've been talking. So maybe I can read them out and say who they were from, and then you can answer them. Some of them, of course, may have been partially answered as you went on forth, but we just sort of see how we go. So the first one from Abhishek asked, does the liberated chlorine have any adverse effects in the immediate vicinity of the mineral accretion technology device?
Okay, chlorine. I'll come back to that diagram again. Yeah. Okay, the chlorine that is liberated, I don't think so. As I pointed out, that hydrogen ions can combine with chlorine to produce hydrochloric acid that will dissolve carbonate. But at the same time, chlorine has high affinity for sodium as well, and sodium chloride is the most abundant salt in the sea. So I don't think that directly chlorine has any problem associated with what was the question on the corals?
Oh, just in the immediate vicinity. So I suppose that would mean,
Immediate vicinity,
corals
yeah.
on
So
the
it
actual
could have,
structure. Yeah.
if hydrochloric acid is built up, it could have some damaging effects. But that is why I said that anode needs to be placed at a distance away from the cathode. Yeah.
Okay, great. Good answer. That makes sense. Great. Another question, also from Abhishek. Curious to know why Benedetti et al. Assessed soft coral settlement on accreted COCO3. Was it an assessment of how accretion affects associates of hard corals?
Sorry, I didn't get that question. Of soft corals.
Yeah, so they were wondering why the study you cited, why there was a focus on soft corals. Was it an assessment of how accretion, mineral accretion, affects associates of hard corals?
No, so they conducted this study on this species specifically. It's nothing related to soft corals as such, but this species, Corallium rubrum. Corallium rubrum is a highly valuable species in the field of medicine. It has high amount of bioactive materials. So they were trying to predict the efficacy of using Biorock technology on growing Corallium rubrum, this coral species on an industrial scale, and why they used accreted calcium carbonate without polarization. So this is a common practice with the practitioners of Biorock. What they do is they basically keep two controls. One of the control is a natural calcium materials. In this case, it was marble and an accreted calcium carbonate without polarization, without any electricity. So they wanted to check whether electricity has an influences the settlement of the larvae, or is it just the calcium carbonate that is influencing it. That is the reason they use two types of this thing, accreted calcium carbonate, one with electrical stimulation and one without it. I hope that answers its question, Abhishek.
I think so.
This is a very common practice.
Yes, yeah. In the group chat, they said thanks. Awesome. So
sorry to interrupt you again. Even in this study by Borel et al., there were two types of control used. Again, the same procedure was used. Calcium carbonate accreted, and in place of marble, they placed bamboo, like that. So I did discuss this with Chad, and it is an important issue to take up in the future. We need to figure out whether the accreted calcium carbonate itself is increasing the growth rate, or it is the free electrons available that are increasing the ATP production, and that increase the growth rate of corals, or they are working together.
Okay, awesome. Great. The next question, this is an interesting one that actually I've wondered before in the past too, from Siddharath. Does the technology have any effect on corals spawning? So I'm assuming by that they mean like of corals that are actually on the structures themselves, I assume. Correct me if I'm wrong, Siddharath.
Yeah, so
but
I
is there
did
any
answer
studies
this
looking at that?
question in my presentation as well.
Oh, yeah.
Yeah, so basically none of the research, this is a very important point, none of the researchers have taken it as a parameter, right? They only look at the growth rate. So yeah, here I mentioned the time of first spawning needs to be considered as a parameter, but none of the studies have done that so far, and quite obviously so, because corals take a lot of time to mature, to sexually reproduce. Like for Acropora, it is around in general, for branching corals it is around a year, so that's manageable. But for massive submassive type, it could go for 8 years to a decade or so. Now researchers are under a lot of pressure to publish, right, because that's how they obtain funding, and that might be the reason why they did not use this as a parameter. So we to answer your question, we don't really know. There's no literature available for it.
Still one of the big questions. Okay, cool. All right, the next question from Karen was, can Biorock technology be applied to India, in your opinion? I guess you talked about some of the challenges and limitations. Do you think it could be applied over there?
Well, of course, I think it can be applicable. In India, we are facing some problems of coral bleaching on the east coast especially, And yeah, the Zoological Survey of India has undertaken Biorock projects in partnership with Dr. Guru, and the results will be out soon. What I predict is that it will be successful in India because India is a tropical country. We experience a lot of insular radiation. So using solar panels, it is quite convenient to set up these plants in India in a very sustainable way. And at the same time, it is very cheap as well. So yeah, it can be applicable.
Okay, cool. Great. Oh, this is also relating to, well, the bleaching point you've just mentioned.
Okay.
From Siddharath as well, another one. He said, it was mentioned that with the Biorock technology, the corals are more resilient to and recover faster from bleaching. Is there a correlation between the mild current and retention or recovery of zooxanthellae by the coral?
Sorry, I did not hear the question properly. Could you please repeat it? Sorry.
Yeah, no, of course. So you mentioned that the corals are more resilient and that they recover faster from bleaching, or it's been suggested that that's the case using mineral accretion technology. So is there a correlation between the mild current and the actual retention of the zooxanthellae by the coral? I suppose the alternative being that they bleach the same, but maybe they recover quicker than accreting or, non-Biorock corals.
Okay, so I could not find any correlation between low current density and bleaching, to answer your question. But actually why this happens is because due to the high availability of electrons, as I pointed out, there's a lot of ATP being produced in the zooxanthella as well. In normal cases, what happens is that due to high temperature, sometimes the photosystems where the actually photosynthesis takes place gets damaged, and that is the reason superoxide radicals are formed inside. Now, inside our bodies, we do have antioxidants, but that might not be the case with coral polyp itself, and that is why it ends up expelling the zooxanthellae out of the coral polyp, right? So, and gets bleached. But in this technology, it has been shown that corals grown on Biorock show zooxanthellae that have higher reproductive capacity and higher chlorophyll as well. So they are not bleaching, but there is no correlation as such between the current density and what was the question? Resistance to
the
bleaching.
retention,
Yeah.
Retention of the zooxanthellae.
Yeah.
Okay, great. Wow. Okay, what was the next one? This is a great one. So from Marco. So Marco asks, do you think that it would be possible to restore both corals and seagrass on the same Biorock structure? So doing multiple forms of restoration at once. Do you think that would be possible?
Not in a single grid. Like these seagrasses, salt marshes, etc. are restored inside a Biorock grid. I don't think that is possible because in the end, what corals need for growth is calcium carbonate, and calcium carbonate buildup will end up smothering and killing the seagrasses. But they could be done side by side. So there is a patch reserved for corals, and some distance from it, we can carry out the restoration of seagrasses at a lower voltage, like that. Just an example. Yeah.
Cool. But what about like oysters and corals? Or I suppose they're usually in different temperatures anyways.
No, oysters are also found in tropical countries as well. But why do we? Yeah. I live in a tropical country and I see oysters all the time. But the thing is that why do we want competition between the organisms, right?
Good point. Yeah.
So yes, it could be done.
There's no necessity to it, really.
Yeah. If we want to increase the ecosystem services, I presume that it should not be done. But again, I could be wrong. That's just my opinion. Yeah, no,
that sounds pretty logical. I think this is more of just a curiosity with the mind of, could you do all the things at once?
Theoretically, everything is possible. Yeah, we need to think about the practicals.
Yeah, of course. Okay, and this was the last one that's come through. Second last one. There was just another one that's come through. But first, I'll go from Abhishek again. Can Biorock technology adversely affect the local distribution of corals and their associates? Can it significantly draw recruitment away from much larger patches that aren't electrified? So I suppose sort of like in shortened words, may it, sort of attract coral communities to where, mineral accretion structures are and actually cause a reduction elsewhere potentially? Is there anything in the literature regarding that question?
No, I could find no literature. Yeah, one thing is about the fish diversity that I spoke about. So Jompa et al. In Indonesia saw a higher coral reef fish density around the Biorock structures as compared to the natural reefs, but there were no inhibiting effects on the neighboring reefs. I found no literature pertaining to that. And it's also the fact that the spatial scale of these projects is quite less, a few meter squares. That is not enough to hamper the growth of a larger reef, a natural reef, I suppose.
Yeah. Okay, cool. Alrighty. And then it was just one more that's come through whilst we've been doing these questions, which was, oops, I've just lost it. In regards to the length of their studies, is it possible that the results being drawn may be misleading as they have been conducted over relatively short periods of time? Which I guess is what you sort of touched on before with the bias towards the fast-growing corals like Acropora.
No, I guess I think he is related to the lower temporal scale of this is related to the lower temporal period. So yeah, actually they do introduce a lot of bias, because any coral, like the coral might show an increase in the growth rate, better health in the beginning, but that might not be the case after a couple of years, since one of the study showed that the effect of accretion stops over the long run, because the accreted mineral inhibits electricity, and then interaction between the anode and the cathode completely stops. Now what happens after that? We don't know. That is why longer studies are necessary. And yeah, this is indeed a bias.
Yeah.
And I presume that you at Kota are carrying out longer studies with longer durations.
Yeah, in the process.
Yeah,
but,
yeah.
But takes
so
a long time.
Some exciting publications are expected, I assume.
Yes. Stay tuned, everyone.
Yeah.
I guess that's all you can say when it's going to take a long time. Yeah, fantastic. Is there anyone else out there that has any more questions? There have been some really awesome ones. Alyssa says, thank you for the thorough information and presentation. Yeah. And thanks for being a good host as well. Thank you, Alyssa, for joining us. I agree with her fully. I think that was really, really great. Maybe we'll give it like another minute or two in case anything thinks of any additional questions last minute that they want to ask. But otherwise, I just want to say thank you again to you. What a wonderful presentation, really, really informative. I'm sure that some of the people that didn't make it or that are asleep right now will be really excited to view this, and I'm sure Chad will be as well. We do have another question that's just come through. Do the anodes need to be replaced over time? And if so, what's the typical lifespan of a titanium anode, for example? And then we've also got some other messages saying thank you. Of course, Pete, everyone, if you need to head off or anything, there's no pressure to stay. We are recording anyway if you want to pick up on last questions.
Okay, I think these are sacrificial anodes. So yeah, I do think that they need to be replaced. But I don't know the lifespan of a titanium. I do not remember it. Titanium anode. It usually lasts longer than others. That is what I can tell you.
Okay. Fantastic. All right.
But if you are interested, I can share a publication about anodes. Yeah.
Just okay. Actually, maybe would you, if you're interested in or okay with that, maybe you could even put your email address in the link below, just in case anyone does have any questions that they think of over the next day or so.
Yeah, I can just type it out in the chat.
Awesome. Great.
There's also some cool stuff about anodes, like titanium anodes give best results because the accreted minerals accrete minerals more rapidly. At the same time, the ratio of brucite, that is magnesium hydroxide and calcium carbonate, is quite less. So we get more proportion of calcium carbonate as compared to magnesium hydroxide, and that's exactly what we need for these studies, but the mechanisms are not very clear as to why that happens.
Great. Oh, so many lovely messages coming through. Thanks to everyone for
Thank you.
joining. Awesome. Okay. Really, 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 really great literature review. I can see what Chad's been talking about.
Thank you.
No worries. Okay.
All
well, in that case, yeah, maybe we'll wrap it all up. Yeah, thanks you for everyone, Vighnesh, for presenting. It's been really great. And let us know if anyone's after any other additional presentations that we can try and organise. But otherwise, yeah.
Okay. Thank you very much for attending. Yeah. Bye.
All right. Thanks, everybody. Have a great morning or evening, depending on where you are.
Bye. Yeah.
Bye.